So, yes, till our previous class we are done with prokaryotic cell, we are done with differences between prokaryotic and eukaryotic, introduction of plant cell and animal cell, we are done with cell wall and fluid mosaic model, cell membrane, functions of its cellular transport. Till there it is done. And endomembrane system, endoplasmic reticulum we have started. Okay. So, today we will once start over again with endomembrane system, and from endomembrane system we will be covering up endoplasmic reticulum, Golgi, lysosomes, vacuoles, and double membrane bound organelles we will look separately in tomorrow's class. Today we will look into the cytoskeleton and centrioles and centrosomes, Cilia Flagella. This is the area we are going to finish off. Okay. So, first when we have to start on with endomembrane system, it reminds us about a teamwork, right. Why only these organelles are placed in endomembrane system? You told me that it is because of their work in a coordinated manner. They function in a coordinated manner, and all the organelles which are placed in the endomembrane system are single membrane bound or double membrane bound? Single membrane bound organelles. Good. Of which the very first organelle is endoplasmic reticulum, second Golgi bodies, third lysosomes, and fourth is vacuoles. In animal cells the presence of vacuoles is very limited to the cells. Okay. So, in animal cells you can limit it up to here itself, in plant cell it is up to vacuoles. Got it? Now, first we learn on endoplasmic reticulum. We have learned it yesterday itself, once again we will recall. Where do you find this endoplasmic reticulum and in what shape do you find? They are network of this reticulum structures, okay, network of tubular structures in reticulum, okay. They are the extensions of what? Nuclear envelope. Nucleus has 4 components we have learnt. Nucleus has what? Nucleoplasm, nucleolus, nuclear pores, nuclear envelope. From this nuclear envelope, they are extending like this into the tubular extensions and forming a network of reticulum like structure. Okay. This network of reticulum like structure will divide the cytoplasm into 2 compartments. What are they? Luminal compartment and extraluminal compartment or space. It is luminal and extraluminal space. They are differentiated based on the surfaces they have. Okay. If at all the surface is rough, that is because of ribosomes attached to it. That is known as what? Rough endoplasmic reticulum. Once we will recall about ribosomes. Ribosomes are composed of RNA and proteins. Remember, ribosomes are composed of RNA and proteins. How many different types of proteins are present? 80 different types of proteins are present. Okay. And then in prokaryotes though we have learnt 70S ribosomes are present, whose larger subunit is 50S, smaller subunit is 30S. Here in eukaryotes you can see in cytoplasm or attached to ER, what type of ribosomes are present? 80S ribosomes. What are they? 80S ribosomes, whose larger subunit is 60S, smaller subunit is 40S. Got it? Which subunit is attached to the layer of the endoplasmic reticulum? Larger subunit is attached to endoplasmic reticulum. By what protein? Ribofourin protein. That is attached by a ribofourin protein. Clear till here? And RER pure function is what? Protein synthesis. This itself you remember. Protein folding also happens, where in biomolecules you have learnt, right, in proteins 2D structure, beta helix, beta sheets and alpha helix of 2D structures, tertiary structures of protein. Such foldings are also possible in the rough endoplasmic reticulum, and then they will be transported in the form of vesicles to what? Golgi bodies. Coming to smooth endoplasmic reticulum, which is not having any ribosome layers over it. okay, and most of the smooth endoplasmic reticulum will be of tubular structures like this, okay. They will be close to area, they do not have any ribosomes. What is the chief function of smooth endoplasmic reticulum? Lipid synthesis, detoxification of drugs later, and it is involved in the synthesis of steroid hormones in an animal cell. In an animal cell, it is involved in the synthesis of steroid hormones, okay, and it will regulate the calcium ion synthesis. Its concentration is being regulated, which controls muscle contraction and relaxation, okay. This will control muscle contraction and relaxation. That smooth endoplasmic reticulum which is present inside the muscle cells or muscle tissues is known as what? Sarcoplasmic reticulum. Sarcoplasmic reticulum, okay. That SCR which is present inside the muscle cells is known as what? Sarcoplasmic reticulum, which controls what? Calcium ion concentration. So, with respect to functions, in RER is only one function, that is protein synthesis, you have to remember. SCR you have to remember, lipid synthesis, detoxification of drugs, very good, calcium ion concentration will be regulated, and if it is in muscle cells you will call it as sarcoplasmic reticulum, very good, and then calcium storage, okay. In carbohydrate metabolism also it is involved, but this will be an extra point to you, no problem, you note down, okay. It is also involved in carbohydrate metabolism. For example, you ate so much of good food, okay, like whatever food you like, might be sweets or biryani or whatever you want, you have ate. Now your cells are equipped with so much of carbs. That carbs will be stored in the form of glycogen. Glucose should be converted to glycogen. There what metabolism is activated? Glycogen metabolism. So for that also which organelle is involved? Smooth endoplasmic reticulum. This is involved in glycogen metabolism, which will control glucose, which will convert glucose to glycogen. You can add on this one more point, okay. So this is all the functions about endoplasmic reticulum. Clear now everybody? Yeah. Now, so these are the basic differences between smooth endoplasmic reticulum and rough endoplasmic reticulum. Can you see everybody? These four differences, so you have to keep it in your mind now. Ribosomes are present, here ribosomes are absent. Appearance is rough, here it is smooth. This is involved in protein synthesis, lipid, detoxification, calcium storage and carbohydrate metabolism, SCR. Major locations, this is a new point you have to take down. RER is abundantly present as in the protein secreting cells. It is abundantly present in what cells? Protein secreting cells like pancreatic cells and plasma cells. Got it right, ma? Next, smooth endoplasmic reticulum is present in all type of cells. Now you tell me, as it is involved in lipid synthesis. It is mostly present in adipocytes or adipose tissue and steroid secreting cells and liver cells, because it is also involved in glycogen metabolism. Okay na? Clear, right? So here what are the few points, new points that you can take down from the previous classes? 1. Carbohydrate metabolism of SCR, and 2. Where they are present in huge number. Okay? Drink some water. Done, right? Shall we move to Golgi? Yeah. So yes, from Golgi bodies, first let me present you in the form of a small cartoon diagram so that you will remember it well. So if I have to suppose this as the nucleus, and this is the envelope and small pores over the nucleus, nucleolus and nucleoplasm, okay. The extensions of it will be endoplasmic reticulum, like this, few extensions, okay, and few tubular structures. Over this ribosome is present, larger subunit is connected, right? 60 years, and from here lipids are being synthesized. Let us suppose those are being secreted in the form of what? Vesicles, be it proteins or lipids. They are secreted in the form of vesicles. Those proteins and lipids which are being synthesized here are naive. They are naive. Okay, means they are not metabolically active. They are not being used to perform the metabolic functions. Now this has to be processed where? In Golgi bodies. In Golgi bodies, which phase of Golgi will be closer to the endoplasmic reticulum? Very good, cis phase. Golgi bodies exact location in the cells, Golgi bodies are densely stained disc shaped structures which are close to the nucleus. They will be close to the nucleus, and cis phase will be towards the endoplasmic reticulum. This is addressed as cis phase or forming phase or convex phase. Got it? Cis phase, forming phase or convex phase. In animal cell, if you take it, in animal cell you can see interconnected cisternae. These structures, these disc shaped structures are known as what? Cisternae. Okay, they will be interconnected in an animal cell like this. Interconnected cisternae you will see. And inside the Golgi bodies, what actually happens? Processing, packaging happens. Golgi bodies are the major sites, or this you will say Golgi complex, Golgi bodies or Golgi apparatus, inside which processing, packaging and transport take place. Processing and packaging occurs inside this Golgi bodies. Means what? The proteins will be glycosylated here. Proteins are subjected to what? Glycosylation. And glycosidation of lipids. Fine, right, ma? Glycosidation of lipids and glycosylation of proteins. In simple terms, to understand what actually happens, glucose molecules or sugar molecules will be adjoined with what? Lipids and proteins inside Golgi bodies. Okay, means like mannose might be, fructose, glucose, okay, will be adjoined with what? Proteins and lipids. That process is known as what, ma? Glycosylation of proteins and glycosidation of lipids. That is how they are processed and packed. From transphase. Transphase is also known as what, ma? Concave side. Transphase, also known as concave phase or concave side, and this is addressed as maturing phase. Trans, concave side or maturing phase. From the concave side, the processed molecules will be again released into the cytoplasm like what? Vesicles. Okay. Likewise, few molecules which are known as glycoproteins and glycolipids. After glycosylation of proteins, what are formed? Glycoproteins. And glycosidation of lipids will result in the formation of glycolipids. They will be sent to plasma membrane, and they will increase the surface area or fluidity of the plasma membrane. Okay, they are transferred to plasma membrane. Golgi bodies will play key role in the synthesis of cell plate or the formation of middle lamella. We have learnt at the time of cell wall formation, right, where the first phragmoplast is synthesized, which is having high concentration of calcium pectate and magnesium pectate, that is being synthesized from Golgi. These two activities you have to remember. Glycoproteins, glycolipids sent to plasma membrane. In plant cell, cell wall formation is taken care by Golgi bodies by synthesis of phragmoplast, which is rich in calcium pectate and magnesium pectate, rest vesicles which are being sent. So, all the molecules which are being transferred from the trans phase has intracellular targets and intercellular targets, yes or no? Means inside the cell and to the outside the cell also they are being transporting the molecules. Those vesicles which are rich in the hydrolytic enzymes secreted from the trans phase are known as what ma? Lysosomes. Okay. Before learning on lysosomes, once let us see the gist. Who discovered it? Camillo Golgi. Year is not required, just remember the scientist name. Likewise, endoplasmic reticulum is being discovered by K.R. Porter. K.R. Porter. And Golgi bodies from Camillo Golgi. Okay. And location exactly near to the nucleus. It is near to the nucleus. What are the major functions? Processing, packaging and transport of proteins and lipids. Modification of proteins and modification of lipids take place here. And if you see the structure clearly, is it visible? If you see the structure clearly, yeah. This consists of flat disc shaped sac like cisternae whose diameter is, each cisternae is how much diameter? 0.5 micrometers to 1.0 micrometer. Important to remember ma. Next cisternae, they are stacked like this. Now I am going to tell you different type of Golgi bodies which are present in different cells. Okay. Stalked type of Golgi bodies you can see only in the animal cell. Okay. And varied number of cisternae are present based on the type of cell and functions of the Golgi bodies. Do you think Golgi bodies are present in all the type of cells? No. They are absent in few and present in high amount in few more cells. Let us see where are Golgi bodies present in high concentration. Golgi bodies in high concentration. First, in the root cap cells, Golgi bodies are present at highest concentration. Root will go via what movement? Positively geotrophic, right? So, this Golgi bodies will help in the penetration of root for positive geotrophic movement. Okay. So, Golgi bodies are highly present or they are rich in which cells? Root cap cells. Okay. And in root, if you remember, root cap is at the terminal region, root apical meristem will be at the subapical region. Fine. So, Golgi bodies are rich in root cap region. Second, in the acrosome of human sperm cell, Golgi bodies are highly present in acrosome of sperm cells. Now, let us see where Golgi bodies are absent. In matured RBC, you do not see any Golgi bodies. In sieve tube cells, fine, no, there will be absence of Golgi bodies. And sperm cells of bryophytes and pteridophytes, which are nothing but antherozoids of bryophytes and pteridophytes. Whichever points you feel like they are fresh and new to you, please take down either in the NCERT or in any sticky notes or your running notes. Okay. Fine, right. This is based on the presence and absence of Golgi bodies. And Golgi bodies are disc-shaped structures, but it can be unicisternal, it can be unconnected, it can be interconnected and stalked. Like if you observe yeast cells, you can see unicisternal Golgi bodies. yeast cells have single cisternae. And then plant cells, you observe unconnected cisternae. Known as dictyosomes. Next, animal cells, you can see stalked and interconnected cisternae. Done, right? Clear? Where they are present in high concentration, in low concentration means completely absent, and then unicisternal, unconnected cisternae, interconnected cisternae in stalks. Done till here? Clear with Golgi bodies? Shall we move on to lysosomes? Yep. Here, coming to lysosomes, first, those vesicles which are immediately released from Golgi bodies are known as what? Primary lysosomes. Okay? Primary lysosomes are those structures which are rich in hydrolytic enzymes, and those hydrolytic enzymes, can you name a few hydrolytic enzymes? Here we have so from here the vesicles are being released. These vesicles now they are rich in hydrolytic enzymes, right? Membrane bound vesicles rich in hydrolytic enzymes secreted from transphase of Golgi bodies, addressed as what, ma? Primary lysosomes. This primary lysosome, if it is adjoined or attached with any phagosome, what is phagosome? Digestive particle inside the cell or any biomolecule which is about to digest, or food particle which is there inside the cell is known as what? Phagosome. Food particle or digestive particle inside the cell. Primary lysosome along with this phagosome is known as secondary lysosome or heterophagosome. Okay, this is known as what? Secondary lysosome or heterophagosome. Now, after digestion into the cytoplasm, this will be releasing out some particles, you can say the undigested particles, okay, that remain inside the cytoplasm after digestion. That undigested particles are known as what? Residual bodies. Residual bodies. And in any living organism, especially amphibians, at the time of metamorphosis might be from larval stage to they are entering to the adult stage. Best example, frog, from tadpole stage to adult stage, means that tail has to be removed and it will be turned to an adult body. At that time what has to happen? Cell lysis have to happen. Cells have to destruct. That is known as programmed cell death or autolysis, which happens because of lysosomes. So those lysosomes, primary lysosomes, if they are attached to any dead cell particles, okay, for cell digestion or cell killing, that is known as, those lysosomes are known as autophagolysosomes or autophagosomes, which help in autolysis of the cell. They aid in what? Autolysis of the cell. Best example is metamorphosis of amphibians. Here we will look into, here you see, lysosomes, the cells will digest, these are the cell digesters and recycling centers. Here can you see the diagram? These are the primary lysosomes. This primary lysosome, first step, they will engulf the phagosome or the digestive particles into the vesicles. First is what? Endocytosis. Endocytosis of food particle is known as what? Cell eating, right? So it will first engulf and then it turns forming what? Phagosome, after which what takes place, ma? Fusion. Fusion with the lysosome and then digestion. At last residual bodies will be released after digestion, and then ephagi or exocytosis will occur. This is the process of what? How lysosomes will digest. This is the process that happens with secondary lysosomes. Lysosome attaching with phagosome via endocytosis. After digestion it will be releasing out the residual particles and then exocytosis. What are the other examples for the functions, lysosomal functions? Includes first what? Metamorphosis. WBC destroy bacteria by what? Phagocytosis. And removal of worn out mitochondria by autophagy. Any dead cell particles inside the cell will be removed by what? Lysosomes only. That is why they are called as what? Suicidal backs of the cell. Breakdown of damaged cells after injury is also because of what? Lysosomes. Okay, these are the added functions that you can take. WBC destroy the bacteria, lysosomes will worn out mitochondria, and breakdown of damaged cells after injury. Lysosomes are discovered by Christiana D. Dewey. He is the scientist, Christian D. Dewey. These are the functions that you already know. They are the suicidal backs of the cell, help in defense and immune system, autolysis, autophagy, heterophagy, and intracellular digestion. These are the 5 functions that are performed by lysosomes. Done, right, ma? With the functions of lysosomes. Can you name few hydrolytic enzymes that are present in lysosomes? Any few hydrolytic enzymes? Lipases, proteases, carbohydrases, hydrolases, nucleases. These are different types of hydrolytic enzymes that are present. So now we are clear with lysosomes as well, right? Shall we move on to vacuole? So next is vacuoles. Vacuole which is present in the plant cell is known as sap vacuole. Why? Vacuole present in the plant cell is known as sap vacuole. Yep. So first tell me, vacuole occupies how much percentage of plant cell volume? 90% of plant cell volume, and the membrane of vacuole is known as what? Tonoplast. Okay. Do you think the concentration of vacuole is less than cytoplasm or more than cytoplasm? More than cytoplasm. Why? No, it is not because of cell volume. Very good. It is because of the molecules that are present inside the vacuole. See, from cytoplasm now we have seen lysosomes are performing so many activities, and all the constituents are present where in cytoplasm they are sent to vacuole, because of which vacuole in a plant cell is filled with sap, excretory products, ions and water, which makes the concentration of vacuole very high. So it has high concentration. Okay, in vacuole the concentration is high. Whereas in cytoplasm, if you see, it has low concentration of the molecules. Transport of any molecules from low concentration to high concentration is what? active transport, right? So to pump the molecules or to transport the molecules from cytoplasm to vacuole requires ATP, and it is also through facilitated diffusion sometimes. You remember, this is via active transport, and this requires what? ATP. This requires ATP, and the movement is facilitated via active transport. This is across concentration gradient or against concentration gradient? Against concentration gradient. Clear, right? Now, we have different types of vacuoles also, you have to remember. Sap vacuole is the one which is present in a plant cell. Likewise, you have contractile vacuole, which is seen in most of the freshwater protozoans. This mainly helps in osmoregulation and excretion. Okay, it mainly helps in osmoregulation and excretion. Next is food vacuole. Food vacuole is present in protozoans. Best example can you give? Ciliated protozoan. Very good. Paramecium. We can see food vacuole that mainly participates in engulfing of food material. Okay, this is involved in what? Ingestion of food material. Next is gas vacuoles. Gas vacuoles are present in prokaryotes also. We have learned when we are learning inclusion bodies as well. Okay, they are present in prokaryotes and they are majorly involved in what? Buoyancy. Example, you can take cyanobacteria and some other prokaryotes. Fine, right? So along with sap vacuole, please remember contractile vacuole, food vacuole and gas vacuole. Clear with this, right, ma? This is the exact composition of cell sap. In NCERT it is given like water, sap, excretory products, ions and water. Water, ions, sugars, organic acids, amino acids, some pigments like anthocyanins, toxic substances and metabolic waste. All these are present in sap vacuole. Please take pigments like anthocyanins, sugars, organic acids, ions and water. Composition is important, ma. The pH of vacuole is around 4.5 to 5.5 concentration. pH is around 4.5 to 5.5. Sap vacuole pH. Right? These are the major functions of plant cell vacuole. It helps in storage of water, for osmoregulation, which keeps the cell turgid. Thereby it also helps in maintaining the turgor pressure and providing the mechanical support to plant. It also helps in detoxification as it is consuming all the waste from the cytoplasm. It also has some pigments like anthocyanins. It is also helpful in pigmentation of the plant cell. Key functions and types, important, ma. And what is the pH of the vacuole, sap vacuole? And how the molecules are transported via active transport. Simple, right? Functions of sap vacuole you have to remember, how it is transported, what is the pH of the vacuole, what are different types of the vacuoles you have to remember, okay, and what are the major functions of plant vacuole. That's all. Clear with vacuole also, no? So this completes what? Endomembrane system. Once a quick recap, sit straight about endomembrane system, sit straight all. Just tell me how much ever you recall, okay. First we have started with what? Endoplasmic reticulum. Two types, what are they? RER and SER. Say it in shortcuts only, no problem. RER has what subunit attached to it? 60S. Via what protein? Riboforin. Okay, it will be in synthesizing only one. What is that? Proteins. Whereas SER has multiple functions we have learned. What are they? Lipid synthesis, detoxification of drugs, calcium ion concentration, And SCR present in muscle cells known as sarcoplasmic reticulum, very good. And that will aid in what? Regulating calcium ion concentration, will help in muscle contraction and relaxation. And after this we have learnt? Very good, glycogen metabolism. These are the 4 functions we have learnt: lipid, steroid synthesis, calcium ion concentration, carbohydrate metabolism. In simple terms, glycogen metabolism, okay, which will reduce the glucose levels, blood glucose levels, converting it to glycogen. Where are the RER present in high concentration? Which type of cells? Whichever cells have high protein synthesis, we have learnt, right? Cells, high amount of RER, high amount of SCR, we have seen adipocytes, liver cells. RER we have seen pancreatic cells. Likewise, okay, I have made you people to write also. If you haven't take down, please take down, that is important. This region, pancreatic cells, plasma cells for RER, adipocytes, liver cells, SCR. Okay, then we have looked into Golgi bodies. Golgi bodies, which phase of Golgi is towards the nucleus? Cis phase, towards the endoplasmic reticulum. What is it receiving? Naive proteins and lipids in the form of vesicles. Golgi bodies are unconnected in what type of cells? Plant cells, known as what? Starts with D only. Dictyosomes. Unconnected Golgi bodies seen in plant cells are known as what, ma? Dictyosomes. Interconnected Golgi bodies which are stalked are known as what? Interconnected Golgi bodies are seen in animal cells. Unicisternal, single cisternae is present in yeast cells. Got it? So this is about different types of Golgi. And now you tell me, where do you see high concentration of Golgi bodies? Root cap cells and acrosome of sperm cells. Next, where do you see less concentration of Golgi or absence of Golgi bodies? Matured RBC, sperm cells of bryophytes and pteridophytes, sieve tube cells. These are the areas where Golgi bodies are absent. Clear with Golgi bodies? Single cisternae is of how much diameter, ma? If you remember, 0.5 to 1.0 micrometer in diameter. Next, ER is discovered by K.R. Porter. ER is discovered by K.R. Porter. Next, lysosomes. Primary lysosomes are rich in what type of enzymes? Hydrolytic enzymes. Their concentration is high, right? And tell me, if primary lysosome is adjoined to any phagosome or digestive particle, that is known as what? Secondary lysosome, also known as heterophagosome. What is the process of digestion? First is endocytosis. Lysosome will engulf that digestive particle. Digestion takes place, okay, and that forms out what? Phagosome. After digestion, that will be releasing out what? Residual bodies, okay, and then that will be sent away from the cell or out of the cell, which is known as what? Exocytosis or effagi. Primary lysosomes, along with any, when connected or attached to any dead cell particles inside the cell, known as what? Autophagosome or autophagolysosome. Okay, this is one more type. 4 types of lysosomes you have to remember. Okay, and then autolysis, that helps in activates during what process in amphibians? Metamorphosis. Okay, so done with lysosome, no? Functions, these 5 functions you have to remember. Lysosomes helps in what? Intracellular digestion, heterophagy, autophagy, that is cell lysis, and autolysis and defense. That is the reason lysosomes are known as what? Suicidal bags of the cell. Last is vacuole. From vacuole, first sap vacuole only we will learn. Sap vacuole is composed of what? Sugars, organic acids, ions, pigments like anthocyanins, very good. Toxic materials, okay. Thereby the concentration of sap vacuole is very high. How much % of vacuole is present in a cell? 90% of plant cell volume. It is surrounded by what membrane? Tonoplast. How the molecules are being transported from cytoplasm to vacuole? Active transport. That is facilitated via ATP. It requires the energy ATP as it is against the concentration gradient. And then we have learnt on different types of vacuoles. Examples, beta? Contractile vacuole, present in freshwater protozoans. Activity of contractile vacuole important. Osmoregulation and excretion. Then food vacuole, present in most of the protozoans, like best example paramecium, engulfing of food material. Next is what? Gas vacuole, present in prokaryotes like cyanobacteria. Got it? And gas vacuoles will help, major function is what? Buoyancy. This is all about endomembrane system. Then we have to start on double membrane bound organelles, but we will do it in a special class again. Now we will look on to non-membrane bound organelles present in the animal cell, of which ribosomes we have already covered. Ribosomes are already covered. Next is centrosomes and centrioles. Okay. So listen carefully, ma. Just 2 minutes. Listen carefully. If I have to suppose this as a plant cell, in plant cell cytoplasm, centrosome is surrounded just like a unit layer. There is no membrane over it. And a single centrosome will have two centrioles as cylindrical structures present perpendicular to each other like this. How centrioles are there? Two cylindrical structures perpendicular to each other. And this centrosome is also known as diplosome. Centrosome also known as what? Diplosome, as it has two centrioles arranged perpendicular to each other. This is not surrounded by any membrane, but the region where centrosome is there is known as what? Centrosphere. This is known as centrosphere. The region is known as centrosphere. What is its role in the animal cell? Very good. Louder, Skanda, louder. Formation of spindle fibers or spindle apparatus. Very good. So centrosomes will help in what? Formation of spindle apparatus. Spindle apparatus will play key role in cell division time. Okay. At the time of centromere splitting, the spindle fibers or spindle apparatus will play key role, and those are being synthesized from where? Centrosome. Right. So this aids in what? Spindle apparatus formation. Spindle apparatus formation. To make it more easier for your realistic view, okay, what is the phase where centriole will duplicate? Centriole duplication happens in which phase? If you remember, very good, S phase. Okay, in S phase centriole duplication will happen. That means a single centrosome will duplicate into two. Do you remember, in S phase DNA duplication happens inside the nucleus, centriole duplication happens inside cytoplasm. Likewise, two centrioles are formed like this. Okay, and at the end of prophase, if you remember, karyokinesis first stage is what? Prophase. At the end of prophase, these two centrioles will move to two opposite poles. These two centrioles will move to two opposite poles, and these are known as what? Asters. Okay, known as what? Asters. These asters will radiate out spindle fibers. Got it right, ma? And you have to tell me which phase it is. Very good. How can you tell that? All chromosomes are aligned to one plate, that is equatorial plate. As centriole's major function is formation of spindle apparatus, if I tell this, you will remember that easily. So I am telling you this again. So what we have discussed? Centrioles present inside the centrosome. One centrosome has how many centrioles? 2 centrioles. Arranged in what way? Perpendicular manner. Likewise, centriole duplication happens in which phase? S phase. Centriole duplication happens in S phase. Centrosome is also known as diplosome, right? And at the end of prophase, these duplicated centrosomes will move in two opposite poles, radiating out what? Spindle fibers. Now you may get the doubt, ma'am, in plant cells centrioles are absent. Then how this function is compensated? How the formation of spindle fibers are compensated? I told you many times, if you remember, you tell me. Now if I write, you will say, हाँ ma'am. Plant cells, they don't have centrioles, but then they have microtubular organizing complex. Remembered? They have microtubular organizing complex which help in the spindle apparatus movement or formation. Got it? Now, what is this centriole structure? If we have to take this central part, single centriole we are taking and dissecting it, and we are observing it under the microscope. This is the microscopic view. Okay. At the center, there is no microtubule. There is what? Proteinaceous hub. We have proteinaceous hub at the center. Okay, proteinaceous hub at the center. This proteinaceous hub is radiating how many radial spokes? 9 radial spokes. To each radial spoke, there are 3 peripheral microtubules. Can you see here? 3 peripheral microtubules known as A, B, C. These peripheral microtubules have CA connections. One is connected to other via what? CA connections. This has a protein known as nexin. That is how it forms what? A cartwheel structure, cartwheel arrangement. As at the center there is no microtubule, but then peripherally they have 3 microtubules attached to each radial spoke. This arrangement is known as what? 9+0 arrangement. This arrangement is 9+0 arrangement. How many microtubules are present in total in a centriole? Can anybody tell me? In this single centriole, how many microtubules are present? 9 radial spokes, right? To each radial spoke, how many microtubules are there? 3 peripheral microtubules are there. So total how much? Very good. 27 microtubules. Good. So this is the story of centriole and centrosome. Here what you have to remember? Their duplication happens in which phase, what is their role, how the spindle apparatus are being moved, and what is this cartwheel organization. Right? Huh. Clear till here? Shall we move on to cilia and flagella? Yeah. Cilia and flagella. So, we have learned when we are learning the motile structures of a prokaryotic cell that prokaryotic cell will have only one motile structure. What is it? Flagella. Flagella is made up of what protein? Flagellin. Likewise, eukaryotic cells or protozoans will also have the motile structures. Flagella, cilia, these are the motile structure. It also has flagella and cilia. If I have to take any flagellated protozoan which doesn't have cell wall, it only has what? Cell membrane. But then the components of flagella are same. It has what? Basal body, hook and filament. This central part is known as what? Axoneme of flagella. Central part or core part of flagella is known as what? Axoneme. If I have to dissect this and see again, here you can see a different cartwheel arrangement again. What is that? At the center you have 2 microtubules. This is known as what? central microtubules. Central microtubules. These central microtubules are encoded with what? A central sheath. They are encoded by a central sheath. To this central sheath, 9 radial spokes are present. At the center, 2 central microtubules covered by a central sheath. To this central sheath, 9 radial spokes are there. There in the centriole, to each radial spoke, we have seen 3 peripheral microtubules. Here you can see only 2 peripheral microtubules, which are named as AB. And here you can see BA connections, connected via an exin protein only. So here you can see 9 radial spokes and 2 microtubules at the center. This is known as 9 plus 2 microtubular arrangement. Right? Clear? And this is known as interdoublet bridge, which is connecting 2 microtubules. Clear, Noma, till here? Now in flagella part, if you see, prokaryotic flagella is made up of a protein known as flagellin. Likewise, eukaryotic flagella is made up of the protein tubulin. And there prokaryotic flagella has 9 plus 0 microtubular arrangement. Here it has 9 plus 2 microtubular arrangement. One more point to be added. Here if we see specifically with respect to components of flagella, hook and filament will have 9 plus 2 microtubular arrangement. Basal body will be having 9 plus 0 microtubular arrangement, because basal body is fixed to the cell membrane here via centriole. Understood? Got it? Basal body fixed to the cell membrane via what? Centriole. So basal body microtubular arrangement will be 9 plus 0. Hook and filament will be 9 plus 2. This flagella is made up of tubulin protein. Okay, and it has what arrangement? 9 plus 0. Central 2 microtubules covered by a central sheath, 9 radial spokes, to each radial spoke 2 microtubule each. So here how many microtubules in total? Total microtubules, 2 central plus 9 radial spokes into 2. Total how much? Done, right? Centriole and flagella cilia. Complete cartwheel arrangement is clear to you, right? Just 2 minutes, go through the NCERT, those 2 paragraphs of centriole, cilia and flagella. If you are having any doubt, please ask me. And then we will be starting with cytoskeleton. If you are okay, if not, tomorrow I will say cytoskeleton. Should I say cytoskeleton? How many interested to listen? Raise your hands. 1, 2. How many are asking me to take tomorrow? Please raise your hand. 1, 2, 3. Okay, majority makes law. We'll do it tomorrow. Because I can see that in your faces. This is the last class. Till here, everything is clear, no ma? Endomembrane system, cilia, flagella and centriole. Done. Thank you.